animal cell, animal cell. Parts and functions of an animal cell. Yeah! Up first, we have the nucleus. Ah, yay! The nucleus is the information center of the cell. It's where all the DNA is stored and all the chromosomes, and it's also where DNA is transcribed into RNA so that proteins can be made. It's a wonderful place to be, the nucleus. If a cell didn't have a nucleus, it would have no purpose in life. The cell wouldn't have any idea what to do with itself. It would just wander on all around the body, pointless, just drifting through its little cell life. Speaking of the nucleus, the endoplasmic reticulum is attached to the nucleus. Wow, what a good transition. The endoplasmic reticulum is split into two parts, the rough and the smooth ER. I only know what the rough does. I have no clue what the smooth ER does. So we're going to talk about that one. As you can see from the picture, the rough endoplasmic reticulum has itty bitty tiny dots on it. Those dots are called ribosomes, which we'll go into more detail later on in this video. But for right now, you just need to know that the rough endoplasmic reticulum, it essentially hides the proteins away from the rest of the cell so the cell won't, try and fight the proteins, or the proteins won't try and just tussle with anything they can find. It essentially kind of shelters them until they've matured through their protein adolescence. And then, once the protein has come out of its little protein butterfly cocoon, it's all beautiful and ready to be shipped off, the rough endoplasmic reticulum wraps it in a little vesicle, a little protective carrier, and it sends it on to the Golgi apparatus, which we're about to talk about right now. If the nucleus of the cell is like a town hall, then the Golgi apparatus would be the postmaster of the cell. The Golgi apparatus receives vesicles from various sites in the cell, such as the endoplasmic reticulum, as we were just speaking about. It puts the protein in a little vesicle, and once it's time, it sends it on to the Golgi apparatus. And what the Golgi does is it takes that vesicle through the cis entrance, and then it'll just store it until it's ready. And then what the Golgi does is it'll take it, whatever was in the vesicle, it'll take it out of the vesicle and it'll form a new vesicle around it. And what this does is it essentially gives whatever's in the vesicle, whether it's like a protein or something, it gives it like its own little unique zip code and like its own little phone number so that wherever it is, it knows where it came from and where it's going. And then the Golgi launches the vesicle out, and the vesicle leaves to the trans entrance, and then it goes to the plasma membrane where it'll just zoop right on through and go wherever it needs to go. Now we've arrived at the lysosome. As you can see, the lysosome is the circular little thing right there. I've circled it. It's circular, circles. The lysosome is like that one weird guy who he wants to help out, so he goes around and he chews up everyone's food for him just so it's easier for them to digest. It's like he's a good guy. He's just kind of weird. So the lysosome is like the trash master of the cell. It's in charge of eating up and just digesting down just damaged organelles or just foreign particles that enter the cell or just random things. It essentially is what it does. It goes, it finds something floating, it'll absorb whatever it wants to just completely destroy and obliterate. And then it breaks that down, and then it sends what it broke down off in its own little vesicle to be used as food and energy and all that fun. So you're probably thinking to yourself, Bill, how does the cell power itself? It does all these amazing, fascinating things, but where does it get the energy? Well, I have the answer for you. It's in the mitochondria. The mitochondria, or the mighty mitochondria, is the powerhouse of the cell. It's the site at which cellular respiration occurs. And it produces ATP, the energy currency of the cell. It's also speculated that because the mitochondria has its own mitochondrial DNA and its own plasma membrane, that the mitochondria was once its own organism that was then absorbed into us. We talked about them earlier, and here they are again. It's ribosomes. Yay! You're probably asking yourself, Bill, what are ribosomes? Well, I'm about to tell you. Ribosomes are how proteins are made. It's quite fascinating. DNA transcribes itself into mRNA, which then leaves the nucleus and either goes to the endoplasmic reticulum or a free ribosome that's just floating in the cytoplasm. Depending on where it goes, the protein will serve a different function. If it goes to a free ribosome, then that protein will be used in the cell for cellular activities. But if the ribosome is produced in the endoplasmic reticulum, then it goes from the ER to the Golgi apparatus, where it then exports the protein to some other part of the body that needs it. Speaking of free ribosomes in the cytoplasm, what's the cytoplasm? Well, I already showed a picture that had the cytoplasm in it, so I'm just gonna go ahead and recycle that same picture. The cytoplasm is the thick, gooey substance that the cell and everything in the cell floats in. It's quite simple. Even a monkey could get it. Well, that wraps up this episode of PBS Kids Special Biology. It was a fun time hosting. I'm Bill Nye. See you kids in hell.